Yeah. Hello, dear colleagues. Can you hear my screen?
Yes.
Yeah. Okay. Greetings from Kazan too. And today I present not a completed study, but some interesting idea that has been confirmed by the means of computer modeling. This idea was born in our small group, and Tatiana Grigorieva is responsible for protein target and biological aspect of the work. I am responsible for synthesis and other chemical aspects of our study, and our Vyacheslav Tribulovich, he monitors thoroughly the way in which our idea is developed correctly. So let me begin with some introduction of my topic. I begin with multiple drug resistance, MDR. It is considered the main cause for a decrease in the efficiency of anti-cancer therapy, the development of relapses, and the tumor metastasis at different stages of the treatment, which leads to death in more than 19% cases of major tumors. And drug resistance remain one of the deadlocks for the low survival rates of colorectal cancer patients, since it develops in nearly all patients, leading to decreased efficiency of the therapy. And as you know, cell resistance is determined by several group of mechanisms originally inherent in all cells and aimed at protection against xenobiotics and factors which are potentially dangerous for the body's functioning. And in my report, I will only touch on drug resistance associated with drug efflux mediated by ABC transport proteins. The family of ABC transporters is widely represented in a variety of organisms, from bacteria to primates. There are two main groups of functions performed by these transporters in the body. These are export of metabolites and xenobiotics from the body, primarily in the liver and kidneys, and protection of the body as a whole, as well as individual organs and tissues from xenobiotics due to their efflux. Among the variety of ABC transporters, three of them play a critical role in protecting the body. These are multi-drug resistance protein, also known as P-GLYCOPROTEIN or PGP, multi-drug resistant associated protein 1, and breast cancer resistant protein. By regulating of many substances, including clinically important,
Maybe you should
transport
turn off video to have better hearing, because it's sometimes not very good. Turn off video, just present your talk.
It's just problems with connecting.
Yeah. So if you turn off video, it would be better.
Just okay. Okay, then this way. The considered transporter protects the body from metabolites and xenobiotic converted in total the widest range of substrates, from small molecules ions to oligomers, sugars, and peptides. And as you can see, they have a similar structure. They consist of transmembrane domains and nucleotide binding domains that are located within the cell. And further in my talk, I will focus on PGP, but everything I say is also true for other transport proteins of this family. So, since we have resistance mediated by enhanced drug efflux, so when drug is trying to enter the cells, it immediately captured by PGP. And the main strategy to overcome this resistance is prevent PGP from capturing the drug by such inhibitors that could be relatively long retained in the substrate binding cavity of the protein. So the drug successfully reaches the target. And in fact, this approach allows us to temporarily increase the bioavailability of the substrate that rapidly released by the transporter. However, in reality, this strategy turned out to be not so effective as expected, which is demonstrated by the results of clinical trials in patients. Here, mainly showed third generation PGP inhibitors, and some problems related with the passing through clinical trials says that there are obvious obstacles that prevent them to use in clinics. And if you can see, the latest clinical trials are not related with cancer disease. Such PGP inhibitors is now tested in patients with neuropathic pain. So we need another way to deal with such transport proteins. And a few words about another general strategy for combating cancer. This is pro-drug concept. It was developed to degrade some proteins that underline the pathogenesis of a number of cancers, and it is based on the heterobifunctional small molecule, and even not so small, which consists of two main parts connected by the linker. One part is ligand of protein of interest, and another part is ligand of ubiquitin ligase. And this molecule is aimed at targeting the protein of interest to degradation through proteasome. Its mechanism of action is given here. It brings together protein of interest and the YT ligase complex and promotes ubiquitination and polyubiquitination of protein of interest, and that's why these proteins labeled in such a way is directed to the proteasome to degrade. Okay, why we couldn't apply this strategy for membrane proteins? Why not? Because we have some evidence that ubiquitination of PGP is regulate its stability. If you can search in literature more, you can find some evidence that PGP protein expression level could be regulated by ubiquitination. And finally, there are some information that ubiquitination could promote the release of protein, membrane proteins from the membrane. Yes, it's exactly that we need, because in original PROTAC is the final aim is to degrade in the proteasome, and when we consider PGP, we just even not to need to finally degrade. We just need to pull out of the membrane this protein, and in this way it couldn't perform its function, pump out the drugs which we want to stay in the cell. So as you know, ubiquitination of proteins is performed through lysine residues, and the chain of ubiquitins is grown through also lysine residues. And at the first stage, we just look at our PGP and find that there are enough lysine residues, and most of them are located in the intracellular part of the protein. It's okay for us to develop chimeric molecule. Next, since PGP is highly dynamic system, and it constantly changes its state from capturing to releasing, we consider two extreme states, such as inward-facing conformation. It's the state where when the PGP is waiting for the substrate to capture, and outward-facing conformation when it has just released the substrate into the extracellular space. And so they are also called open state and closed state. Firstly, we need to choose which small molecule we will be used as ligase ligand. That is, we need to choose the ligase that will be performed our ubiquitination of PGP. So at this step, we used a pretty tool, GPS Uber. It predicts the possible interactions between ligases and substrates. Firstly, we try to use the most common ligases within the PROTAC concept, that is CRBN and VHL ligases. And for the first, we didn't find any sites of interaction, and for the second, there are a few just sites. So we tried another ligase. It's MDM2, our lovely target. And this tool showed that there are a lot of possible sites for the interaction. So we next took MDM-E3 ligase and check whether it could form complexes with PGP. And so we obtained some complexes with its interaction at the phase H of PGP in the close conformation. Some complexes with interaction around the so-called leg in the case of open conformation, and around the outer surface of nucleotide binding domain in the case of closed conformation too. So next, we came close to the problem of the ligand for the protein of interest. To speak about ligand, we needed to find a site for this ligand, and we used a site map to find some areas to start with, and expectedly we obtained interesting picture that there are some sites within the nucleotide binding domain and one combined site for transmembrane domain. So I want to say a few words about transmembrane domains. So it's commonly used for search and development of PGP inhibitors, but the problem of this domain is consisted in that it is very difficult to use computer modeling to find inhibitors because there are no clarified border between transported substance and inhibitor, and both of them bind to the cavity, but the difference is in the speed of moving through the channel toward formed by two transmembrane domains toward the exit of the cell. And we formulated this conclusion in our paper. So I go next. We left this site and suggested that we can use another site to find inhibitors of PGP. And Alexandra Sagaidaka was talking about this yesterday. I will not go into details of this study, but I want to say that after this work, we get a library of ATP or AMP mimetics that is really exist and which we used to make docking studies for our project. So we used this library for docking and really obtained very quite expected results. Some of the compounds have potent binding to the transmembrane domain in the open conformation. Some of the substances, as they are AMP, ATP mimetics, bind to the nucleotide binding domain within the ATP binding pockets. And some of the compounds have potent binding to the outer surface of the nucleotide binding domain. And this seems to us very interesting because as we can see and we can analyze docking values, we have some special interest to one of the compounds that showed practically the highest docking score to this interesting site, but showed a little interaction with nucleotide binding, ATP binding cavity, and there are some selectivity for binding between transmembrane domains. So now we are working on the generating library of its modification, and so what is interesting that in recent work, some researchers proposed two new sites for PGP inhibitors, and one of them is exactly that we find in our studies. So here are my conclusions. We simulated the formation of complex with MDM2. We identified PGP sites suitable for binding a chimeric molecule and generated primary library of structures with high affinity to this site. And we have a little left to prove our idea. So I just thank you for your attention.